Hello students, today we are going to start learning about a new chapter in NEET Zoology, and that new chapter that we will be learning today is molecular basis of inheritance. Molecular basis of inheritance is the new chapter that we are going to learn from today onwards. Okay. So students, in our previous chapter, Principles of Inheritance and Variation, we have learnt about the pattern of inheritance and also the genetic basis for the pattern of inheritance, right? So, in this chapter, Molecular Basis of Inheritance, we are going to learn about the genetic material. We will be learning about the genetic material in this chapter, Molecular Basis of Inheritance, okay? So, students, we have already come across this word nucleic acids, right? Nucleic acids. Nucleic acids are actually of two types, okay. Living organisms contain nucleic acids. Nucleic acids are of two different types, okay. They are ribonucleic acids, which is RNA, and deoxyribonucleic acid, which is DNA, okay. So, students, DNA is the genetic material. DNA is the genetic material for most of the organisms. Okay, DNA is the genetic material for most of the organisms. DNA is the genetic material for most of the organisms. Okay. And RNA is the genetic material for a very few organisms. For a very few organisms, it is a genetic material, but in all the other organisms, it acts like a messenger or a structural or a catalytic molecule. Structural or a catalytic molecule, okay. So, here in this chapter we will be learning in detail first about the structure of DNA. So, in today's class we are going to learn about the DNA, okay. So, today's chapter that we are learning is DNA, which is deoxyribonucleic acid. Deoxyribonucleic acid. So, DNA, okay. So, students, this DNA is consisting of many nucleotides. It is a polymer of nucleotides. It is a polymer of nucleotides, okay. It is a polymer of nucleotides. It contains many nucleotides. The length of this DNA is identified by the number of the bases, number of nucleotides present in it. The length of DNA is a polymer of nucleotides. It contains many nucleotides. So, the length of DNA will be identified by the number of nucleotides or the number of nucleotide pairs, which are called as base pairs, okay. We can identify the length of DNA by the number of nucleotides or the number of nucleotide base pairs, the nucleotide pairs which is called as base pairs, okay. Let us see some examples for the number of nucleotides. If you take a bacteriophage consists of 48,502 base pairs, okay. Whereas, if you consider a bacteria called Escherichia coli, E. coli, in shortcut it is called E. coli. E. coli consists of 4.6 into 10 to the power of 6 base pairs. If you consider human beings, in a haploid set of chromosomes, they contain 3.3 into 10 to the power of 9 base pairs. In that way, the number of nucleotides is characteristic to every individual. Number of nucleotides is fixed. Number of nucleotides is characteristic to any individual, okay. So, let us now learn in detail about the structure of this DNA, okay. Let us now learn in detail about the structure of this DNA. we are going to learn now about the structure of a polynucleotide chain, a polynucleotide chain. Okay. So, students, here a polynucleotide chain, it consists of, a polynucleotide chain consists of many nucleotides, and each nucleotide consists of a nucleotide. It consists of 3 components. Okay, it consists of 3 components. The first one is a pentose sugar, Okay, this pentose sugar, if it is in case of RNA, the pentose sugar is ribose. Okay, if it is DNA, then the pentose sugar is deoxyribose. Okay, so the difference is there in sugar molecules. Between DNA and RNA, the difference is there in the pentose sugar molecules. RNA is a ribose sugar and dRNA is consisting of deoxyribose sugar. Okay, and it consists of a phosphate group, and nitrogen base. It consists of a nitrogen base. Okay, these nitrogen bases are of 2 different types. The nitrogen bases are of 2 different types. Okay, they are purines and pyrimidines. See, these purines are of 2 different types. Again, purines are of 2 different types. They are adenine, short formly represented as A, and guanine, which is short formly represented as G. Okay. Pyrimidines, they are cytosine, represented as C, uracil, represented as U, thymine, which is represented as T. Okay. So, these are the 3 different types of pyrimidines. See, students, adenine, guanine and cytosine, these 3 are present in both RNA and DNA. Okay. Adenine, guanine and cytosine present in both DNA and RNA. Okay. But uracil is present only in RNA. Uracil is present only in RNA, whereas thymine is present only in the DNA. Okay, uracil is present only in the RNA, thymine is present only in the DNA. That is the main difference between the nitrogen base of DNA and RNA. The nitrogen base of RNA is uracil, the nitrogen base of DNA is thymine. Okay, the nitrogen base of RNA is uracil and that of DNA is thymine. Okay, so that is the differences between RNA and DNA. Let us see now from all these 3 components how a nucleotide is formed. We have learnt here that a polynucleotide chain contains many nucleotides. Each nucleotide is formed up of 3 components. They are pentose sugar, which is in case of RNA ribose, in case of DNA it is deoxyribose, and contains a phosphate group, a nitrogen base, which these nitrogen bases are of 2 types, purines and pyrimidines. Purines, adenine and guanine are the purines, represented as A and G. Pyrimidines are of 3 different types, cytosine, uracil and thymine. Okay. So, adenine, guanine and cytosine are common in DNA and RNA, whereas uracil is found only in RNA and thymine is found only in DNA. This is the difference in the this is the difference in the nitrogen bases of a DNA and an RNA. Okay. So, now let us see how these 3 components of a nucleotide are exactly assembled to form a nucleotide. Okay. So, students, here we have studied that there is a nitrogen base, right. So, this nitrogen base, it will get linked to the, nitrogen base, it gets linked to the first OH group of the first carbon of the pentose sugar, okay. Nitrogen base gets linked to the OH of the first carbon of the pentose sugar by N-glycoside linkage, This results in the formation of nucleoside. A nitrogen base links to OH of the first carbon of pentose sugar by N-glycosidic linkage to form a nucleoside. Nucleoside like adenosine, adenine. Adenine is actually a nitrogen base, right? So now it becomes adenosine or deoxyadenosine, depending upon the type of sugar to which it is attaching. Deoxyadenosine or guanosine or deoxyguanosine, cytidine or deoxycytidine, uridyl or thymidine or deoxythymidine. In that way, this different type of nucleosides will be forming, okay. Nitrogen base will get linked to the OH group of first carbon of the pentose sugar by N-glycosidic linkage to form a nucleoside like any of these, okay. Now, the phosphate group, it will get linked to the OH of the fifth carbon of the pentose sugar, okay, of the pentose sugar by phosphoester bond, okay. The phosphate group is linked to the OH of fifth carbon of the pentose sugar by phosphoester group to form a nucleotide, okay, to form a nucleotide of its respective type, okay. And now the two nucleotides will be linked to each other, by a phosphodiester linkage, between 3 and 5th carbon, between the 3 and 5th carbon of the pentose sugars, okay. Between the 3 and 5th carbon of the pentose sugars, there will be a phosphodiester linkage connecting the two nucleotides. It will result in the formation of a dinucleotide. In that way, many 3 to 5 phosphodiester linkages will occur, resulting in the formation of a polynucleotide, okay. Many phosphodiester linkages will result in a polynucleotide. It will result in a polynucleotide, okay. So, let us now look from the picture regarding very clearly how this phosphodiester bonds are formed, okay. So, once again we will revise everything and then we will continue to learn more. The nitrogen base is linked to the OH group of the first carbon of the pentose sugar by N-glycosidic linkage to form a nucleoside. And then fifth phosphorus group is linked to the OH of the fifth carbon of the pentose sugar in the nucleoside by phosphodiester bond to form a nucleotide. Two nucleotides will be linked to each other by phosphodiester linkages between 3 and 5th carbon to form a dinucleotide, okay. In this way, many phosphodiester linkages will be formed and form a polynucleotide, okay. Let us see how these polynucleotides appear. So in this way the nucleotides will be arranged. Okay. So you can observe that here you can observe there is phosphorus linked to one nucleotide, right. Phosphorus linked to one nucleotide, and the other phosphorus, the other carbon of the other nucleotide, they are linked with each other. So this bonding is actually phosphodiester bond. I have written it as PDB shortcut, in shortcut phosphodiester bonds. Okay. Here from this you can observe very clearly that the pentose sugar is there, and here you can observe the phosphorus groups, right, and you can observe these nitrogen bases, right. So you can observe one more thing, that here the pentose sugar and the phosphorus, they form the backbone of the polynucleotide. They will be forming the backbone of the polynucleotide. Okay. Whereas these nitrogen bases will be projecting from this backbone. The nitrogen bases will be projecting from this backbone. Okay. The nitrogen bases, they will be projecting from this backbone. Okay. The only difference between DNA and RNA is, in RNA on the second carbon of the pentose sugar, on the second carbon of the pentose sugar, in RNA on the second carbon of the pentose sugar, there will be an extra OH group. There will be an extra OH group. Okay. Because of this reason the RNA is different from DNA. Okay. So students, the DNA as the first genetic material, DNA was first observed by Friedrich Miescher. Okay. Friedrich Miescher has first identified DNA during 1860. So during 1860s only he identified DNA as the genetic material. Okay. 1869, in 1869 exactly, Friedrich Miescher has identified DNA as the genetic material. Okay. So he identified the presence of acidic DNA inside the nucleus. Inside the nucleus he identified the acidic DNA, but he called this as nuclein. He called this as nuclein, but he failed to isolate this DNA. He actually failed to isolate this DNA because of the lack of proper techniques for isolating of DNA at that 80s, okay, 1800s. In that time there was no proper techniques for isolating the DNA. Because of lack of the techniques for isolating DNA, these Friedrich Miescher couldn't isolate the DNA. But later on in 1953, there came two people called as James Watson and Francis Crick. These two persons in 1953, they proposed the structure of DNA. They proposed a structure of DNA. Okay. They proposed a double helix structure of DNA. Structure of DNA. They proposed a double helix structure of DNA, which is the most accepted structure, which is the most popular structure of DNA. Okay. So they have proposed the structure of DNA based on the X-ray diffraction studies. Okay. Based on the X-ray diffraction studies which were done by Wilkins and Rosalind Franklin. Okay, by Wilkins and Rosalind Franklin, the X-ray diffraction studies were made. So based on the X-ray diffraction studies of Wilkins and Rosalind Franklin, these people have proposed a double helix structure of DNA. They have proposed a double helix structure of DNA based on the X-ray diffraction studies. Okay. And the very important feature of this double helix structure of DNA which these people have proposed is the proportion of nucleotides. The proportion of nucleotides is a very important concept which was proposed in double helical structure of DNA. Let us learn more about the proportion of nucleotides. Okay. Before that, we will revise whatever we have learned here. A pentose sugar and phosphorus are going to form the backbone of the DNA. Nitrogen bases are projecting from this backbone. The difference between RNA and DNA is on RNA, on the second group carbon of the pentose sugar, there is an extra OH group. Okay. Frederick Miescher in year 1869, he actually identified DNA. He identified the acidic DNA inside the nucleus in 1869 only. He called it nuclein, but he was unable to isolate it and propose its structure because of lack of proper techniques for isolating the DNA at that particular time. Okay. That is why he couldn't do it. But in 1953, James Watson and Francis Crick have come. They have proposed the structure of DNA which is the most popular, most accepted structure, that is double helix structure of DNA. Okay, double helix structure of DNA. So, according to X-ray diffraction studies made by Wilkins and Rosalind Franklin, they proposed this double helical structure of DNA. Okay, now we are going to learn more about this double helical structure of DNA. So, students, as I have told you, the proportion of nucleotides, the proportion of nucleotide base pairs present in the new DNA is very peculiar. Okay, here the proposition of nucleotide base pairs inside the DNA is a very peculiar feature. Okay, so they have proposed that there are nucleotide base pairs inside the DNA. Okay, so this proportion have come actually after the observation made by Chargaff. Okay, observation made by Chargaff. After the observations made by Chargaff, they have proposed the presence of nucleotide base pairs inside the DNA. Okay, Chargaff has observed that the proportion of adenine is equal to thymine and the proportion of cytosine is equal to guanine. The proportion of cytosine is equal to guanine. In that way, he made this observation that adenine is equal to thymine and cytosine is equal to guanine. And based on this observation, they have come to know that these nucleotide will occur in pairs. They have come to know that these nucleotides, they will occur in pairs. Okay. And the another feature that was important about DNA double helical structure was DNA is consisting of two strands of polynucleotides, right. DNA is consisting of two strands of polynucleotides, Okay. So, these two strands of polynucleotides, they are showing base pairing. Okay. These polynucleotides, they show base pairing. Okay. And they are said to be complementary. To each other. Okay. They are said to be complementary to each other. Generally, students, always a purine is pairing with a pyrimidine. Okay, always a purine pairs with a pyrimidine. Always an adenosine pairs with a thymine. Okay, always adenine pairs with thymine. Always guanine pairs with cytosine in the DNA. Okay, always adenine pairs with thymine, guanine pairs with cytosine. And here they have told that there are two strands of polynucleotides which are complement to each other. That means in cell divisions, in cell divisions, one strand of DNA which is considered as parental DNA, one strand of DNA which can be considered as a parental DNA can perform one strand of DNA which is called as parental DNA. It can perform a cell division, it can involve in replication by acting as a template for new DNA, That means according to the base pairs present in one strand of DNA, a second strand of DNA can be produced. The first strand of DNA is acting as a template for the next strand of DNA. By just observing one strand of DNA, a second strand of DNA can be identified. Okay, a second strand of DNA can be produced. So one strand of DNA is acting as a template for new DNA. That means exactly the original kind of DNA will be produced. Okay, so two strands of DNA are present. For example, on one strand there is A, on one strand there is T, one strand there is G, one strand there is C. If you are taking one strand as template, if you are taking A strand and G strand as template, on the other side, as I have told you already, A will pair with thymine, so the other strand T will be produced. Guanine is pairing with cytosine, G is pairing with cytosine. On the other side, C is going to be produced. So these two DNA are same, right? So in this way, this kind of base pairing between the two strands of DNA is important in conservation. It is important in the conservation of DNA, and this is the reason behind the genetic basis of inheritance. Okay, this is the reason behind the inheritance. Okay, because the DNA is not changing. However, it is just passing on. It is getting multiplied in the same way. It is just passing on. Okay, so that is about the importance of this complementary strands of DNA. Okay. Now let us learn about salient features of double helix structure of DNA, double helix structure of DNA. Okay, so salient features of this double helix structure of DNA we are going to learn now. The first thing is DNA consists of two strands of polynucleotides, which are complement to each other, Okay, there will be two strands of DNA which are complement to each other. Okay, and the second feature is here in this DNA. Okay, here in this DNA we will be observing that the two strands are having antipolarity. The two strands exhibit antipolarity. That means one strand will run from 3 to 5 direction and the other strand will run from 5 to 3 direction. Okay, what is this 3 to 5 and 5 to 3? See students, as we have studied regarding the structure of DNA, right? So, how was the structure of DNA? So, this will be how a DNA strand will be appearing like. So, if you say the one side on the fifth carbon on one side there will be a free phosphate group. On the fifth carbon on one side there will be a free phosphate group. So, this side is called as 5' end. Okay, this side is called as 5' end. On the other side you see on the third carbon there will be a free OH group. This is called as 3' end. Okay, this is called as 3' end. So, students, here the two strands are having antipolarity. That means one strand will be running in 3 to 5 direction, the other strand will be running in 5 to 3 direction. Okay. That means if these are the two strands of DNA, this strand will be 3 to 5 and the other strand will be 5 to 3. Okay. In that way, the two strands of DNA will be opposite to each other. They will be running in anti-polarity manner. Okay. Anti-polarity will be exhibited by the two strands of DNA. Okay. That is the second feature of double helical structure of DNA. Then the third feature is their nucleotides occur in pairs. Nucleotides are occurring in pairs. Okay, there is a pairing between the nucleotides. Base pairs are found. Base pairs are found. Okay. So, students, remember these nucleotides are pairing with each other just by using hydrogen bonds, That is why during the multiplication of DNA, these two strands are able to easily get separated and they are able to form a new strand of DNA. Okay. So, using hydrogen bonds, the two nucleotides will be paired up with each other. Okay. So, always adenine, it pairs with thymine, and between adenine and thymine there will be two hydrogen bonds. I am indicating it with two lines. Always between adenine and thymine there will be two hydrogen bonds. Always between guanine and cytosine there will be 3 hydrogen bonds. Okay. Always between guanine and cytosine there will be 3 hydrogen bonds. Okay. Adenine as pairing with thymine by using 2 hydrogen bonds. Guanine is pairing with cytosine using 3 hydrogen bonds. Okay. So, with this you can understand which binding is more stable. Guanine cytosine binding will be more stable. Okay. And you can also understand one more point, that always adenine will be equal to T and guanine will be equal to C, because both of them are in paired conditions only. The number of adenines will be equal to number of thymines, number of guanines will be equal to number of cytosines. Okay. So, that is about nucleotides. Then let us see more features of a double helical structure of DNA. Before we continue to learn more, we will revise this once. DNA is consisting of two strands of polynucleotides which are running complement to each other. The two strands are showing anti-polarity. One strand will be running from 3 to 5 direction, another strand will be running from 5 to 3 direction. Nucleotides always occur in pairs forming base pairs. They are called base pairs. They are paired using hydrogen bond. Adenine pairs with thymine using 2 hydrogen bonds. Guanine pairs with cytosine using 3 hydrogen bonds. So, binding of guanine to cytosine is more stable than adenine to thymine. And always number of adenine is equal to thymine, number of guanine will be equal to cytosine. Okay. Then the next feature of the double helix structure of DNA is, the DNA is actually a negatively charged right-handed coiled. It is coiled in a right-handed direction. Okay. The DNA is not straight. It is coiled in a right-handed direction. Okay. DNA is coiled in a right-handed direction. And each turn of DNA will be having a length of 3.4 nanometers. You know what nanometer means? A nanometer is equal to 10 to the power of minus 9 meters. Okay. So, each turn of the DNA is having a length of 3.4 nanometers. Okay. So, in this way the DNA strands will be appearing. Okay. In this way the DNA strands will be appearing. They will be like they are coiled around each other. If suppose if this is one turn of DNA, this will be measuring 3.4 nanometers. Okay. And in each turn of DNA, there will be 10 nucleotide pairs. Okay. Each turn contains 10 nucleotide pairs. Each turn contains 10 nucleotide pairs. That means length of each turn is 3.4 nanometers. Each turn is containing 10 nucleotide pairs. That means these nucleotides are separated from each other by, they are separated from each other. They are separated from each other by a distance of 0.34 nanometers. They are separated from each other by a distance of 0.34 nanometers. Okay. So, this is one more feature of the double helical structure of DNA. Okay. And you know, the other thing is the plane of all nucleotide pairs is same. All nucleotide pairs is same. Okay, all the nucleotide pairs are lying on the same plane. All the nucleotide pairs are lying on the same plane. Okay, because the same plane in the sense, for suppose like this, A, T, G, C, G, C, like this, all A, T, like this, all the nucleotide pairs will be lying on the same plane. So, this presence of all the nucleotide pairs on the same plane will add stability, will increase stability of the DNA structure. It will increase the stability of the DNA structure. Okay. So, this is one more feature about the DNA double helix structure. Okay. So, these are some of the salient features of DNA double helix structure as proposed by James Watson and Francis Crick. Okay. So, students, this is all about salient features of DNA, and you know, this identification of the structure of DNA has been a very big revolution. It occurred in 1953 and it's been a very big revolution in the science whatever has occurred till now. The very big discovery was the structure of DNA. Okay, and after that, students, now we are going to learn about one more important concept which is occurring in the living organisms. So, very soon Francis Crick very soon has proposed a central dogma, of molecular biology, a central dogma of molecular biology, which is explaining that how is information produced from DNA. Okay, it is explaining that how is this information produced from DNA. So, this is explaining that from DNA information will be passed into RNA, from RNA information will be passed into proteins. So, how DNA is converted into RNA, RNA into proteins, let us learn now. So, DNA, it undergoes transcription and it produces RNA. DNA undergoes transcription, produces RNA. RNA by translation, it produces proteins. RNA by translation, it will be producing proteins. Okay, DNA undergoes transcription, producing RNA. RNA undergoes translation and producing proteins. This is generally happening. From DNA, RNA is produced. From RNA, proteins are produced. This is the general things happening by using DNA. But in case of some viruses, RNA is converted into DNA. Okay, like for example, you see HIV virus. We have already learnt about HIV virus. So, it's a retrovirus, it's having RNA. From using enzyme reverse transcriptase, from RNA, DNA is produced. So, conversion of RNA into DNA is called as reverse transcription. So, in case of some viruses, reverse transcription will occur, converting RNA into DNA. Some viruses, this is seen, reverse transcription is seen. Example, HIV. Okay. So, students, this is all about the structure of polynucleotides. Okay. This is all about structure of the polynucleotides. Okay. Now we will just get introduced to one small concept which we will be continuing in tomorrow's class. Okay. See students, what was the number of nucleotide base pairs found in the human beings? I have told you in haploid set 3.3 into 10 to the power of 9 billion, right, in a haploid set. When we take a diploid set, of chromosomes, then a human consists of 6.6 into 10 to the power of 9 nucleotides, right. So, but if you just consider the distance between the nucleotides, if we are multiplying the distance between the nucleotides and number of nucleotides, then we can obtain the length of DNA, okay. Length of DNA can be obtained by multiplying the number of nucleotides in the DNA into distance between nucleotides, okay. So, now what is the number of nucleotides in DNA? 6.6 into 10 to the power of 9 nucleotides are found in human DNA. Distance between nucleotides is 0.34 into 10 to the power of minus 9. Yes, 10 to the power of minus 9 meters. So, in this condition, if we are multiplying 6.6 into 10 to the power of 9 into 0.34 into 10 to the power of minus 9, the answer will be 2.2 meters. So, the total length of our human DNA is 2.2 meters. The 2.2 meters is the total length of human DNA. But our human cell, generally the size of human cell is actually 10 to the power of minus 6 meters. Okay, 10 to the power of minus 6 meters is the size of human cell, but the length of DNA is 2.2 meters. How this 2.2 meters DNA is fitting into 10 to the power of minus 6 meters cell? This we will be learning in the next class. Okay, so that's all for today, students. And we have, in today's class, we have started to learn about this chapter, molecular basis of inheritance. Here we have learned that the genetic material is DNA. How this DNA is acting like a genetic material, what is this DNA containing, and the structure of DNA. We have salient features of this double helical structure of DNA and other aspects related to the structure of DNA we have learnt in today's class. In tomorrow's class, we will be learning about how this long DNA is packed into such a small cell. Okay, so we will continue our class in tomorrow. That's all for today. Thank you all for joining the class.